So what we represent is one component of SCAR with which most of you may be familiar. It actually looks at the coordinated, global, impactful science research dominantly concerned with Antarctica and Southern Ocean. This is through three groups, that is the geosciences, life sciences, and physical sciences. And this particular series of seminars is related to geosciences. The idea behind this series is to introduce interested people to the activities of the geosciences group to create interconnectivity, and therefore we have requested our action and expert group leaders and associated, hopefully the interdisciplinary groups also to participate in that. So in that series, if we look at the next slide, you see that we had the first seminar. Next please. The on the geosciences group is pointed. The first was on 8th July, and that was on the AdMap, and today it is on IcePro. It will be presented by Johan and Dimitris. Before I hand them over to you, let me introduce us also. The chief officer for the geosciences group is Jesus. The deputy chief officer is myself. Laura is the secretary, and our geoscience fellow is Matilde, who is hosting this meeting through SCAR. Next, please. So we look at that, and this is the second webinar on IcePro, which is essentially looking at regional collaborations as well as methodologies of paleoclimate proxies in relation to the existing situations. And of course there will be a bigger description of that forthcoming. And this seminar is essentially to link us together to answer questions and possibly to take this next to the next level. Next please. So these are the scheduled ones, and there is a simple method to this to register for this seminar, for this series. There is only one link which is there. It is valid for all the webinars, and the registration is through this platform or through this code. Just before I hand over to Johan, one more reminder that next month we are meeting, or many of us will be meeting, in the International Symposium on Antarctic Earth Science, which is the flagship program of the Geosciences Group. Hopefully we will see and meet and discuss some of the things which we are doing here. I hand over to Johan next. Thank you.
Thank you very much, Naresh. I'm going to share the presentations. Can you see it well?
Yes.
Yes.
Okay, so hi everyone. Thank you very much, Naresh and Matilda, for the introduction. So I'm in Joan Etourneau, I'm working at the PHE, and I'm based at the University of Bordeaux in France. I've been working on paleoclimate research in the Southern Ocean for a while now. And since a couple of years, we have been developed this action group, which is named IcePro, which is an international collaboration effort for improving paleoclimate research in the Southern Ocean. So we initiated this project with Carlota and Scutia, and Dimitri Evangelos replaced Carlota recently. And then now we are leading together this action group. So the idea behind this action group is mostly based on some evidences, as we were clearly observing over the last 20 years that in fact most of the data, the paleoclimate records all around Antarctica, were mostly qualitative data, or at best semi-quantitative. So which means that we were still missing robust quantitative data to reconstruct, for instance, temperature change, to reconstruct sea ice variability. So that was the first issue we had to face. And then we also realized that over the last years, we still have some issues regarding the age model that was applied to the different sediment cores. And especially we have very few age control regarding the last centuries or the last 2000 years that are actually used as a baseline of the modern climate. And then while we are working on different proxies, organic proxies, inorganic proxies, we have still some issues about the interpretation of them, mostly because we are clearly missing some calibration. We still do not fully understand, for instance, the distribution of phytoplankton species that are producing the microfossils we are looking at in the sediments. We still don't know why we have this distribution, why we have this species. And then also there is some emerging new proxies, like for instance, DNA, that have been recently developed around the Southern Ocean, and for which we found out that actually we have a few modern data that can be used to compare with the paleo records. So which means that when you look at this map, actually you see that we are also missing quite a lot the most recent years, centuries, or even the last millennia of the Antarctic climates, especially from the marine parts. And then we have this issue about the marine proxies. So the main objectives of ICEPRO is to better understand the past and present mechanisms affecting the Southern Ocean drivers and the biogeochemical composition, including, for instance, the nutrient cycles, as well as the ecosystem structure and dynamics. So for that, our idea is mostly to bring together a wide scientific community from different fields. It could be from geology, from oceanography, from models, from biology, and then to coordinate this effort to provide a robust paleo-proxy calibration. Based on that, we also aim to generate accurate and widely spread paleo records for the last centuries and millennia, and then also closely cooperate with ICEPRO and modeling scientists in order to define as much precise as possible the climatic baseline that will be used for the future projections. So we have a clear strategy that we have defined over the last years. One is the first to improve the coordination of the sampling efforts, mostly because from now there are different national and international programs that are focusing on collecting samples, but clearly there is no real coordination or not so much interaction between these different programs. So the idea is to improve that through different projects and programs, develop new ones when is needed, and including partnership also with private companies. And by this, we mean that, for instance, we can work on some companies that have this kind of monitoring program going to Antarctica from November to March, and then collect a sample that we cannot usually take by using only oceanographic vessels. So it's also a kind of opportunity. Then we want to harmonize the different methodologies in a way that, for instance, when we are collecting surface sediment or when we are collecting water, we want to make sure that we are exactly using the same common and comparable protocol. And this will aid the development and interpretation of paleo records, especially when we want to get a robust calibration for the different paleo proxy from the water column to the surface sediment and including the last 2000 years. And because we can compare these different methods, we also hope to improve the proxy calibration and even maybe reanalyze some samples that have been measured previously, maybe 10 or 20 years ago. And then in the end, we would like to improve the model simulation by providing some new data sets complementary to the existing one in order to have a better comparison or even to assimilate this data into the new model simulation. So in ICEPRO, we have quite a large steering committee involving different partners from different countries. I would say that Carlota Escutia and Andrés Rigual from Spain were almost the first because we initiated ICEPRO through a CSIC project. That was the first step of ICEPRO before it became a SCAR action group. Then we have some partners from France, from US, from Australia, India, Italy, Germany, Japan, New Zealand, UK, and even China. It's not a restricted list in a way that we are also open to invite more people from different nations to join us, even including in the steering committee. But our first step was, of course, to involve these people from different countries in order to cover the largest area possible around Antarctica if we wanted to obtain or improve this proxy calibration and obtain some new paleo-records. In terms of timeline, ICEPRO started in 2022, but like I said before, ICEPRO was even initiated a bit before through a Spanish program. But it's since 2022 until 2026, we aim progressively to build a working group focusing on selected paleoproxies, then to define the common sampling strategy and de-harmonize the analytical work at sea and onshore, which means on board, but also between the different labs involved. Then to identify the vessels sailing the next season to Antarctica, which is a critical point if we want to coordinate the sampling effort around Antarctica. And then since 2024, we progressively summarize our progress made during the previous field season, which may be a bit tricky because, of course, from one season to another, there are some areas that can be reached and some others that cannot be. Then we aim to provide an extensive portfolio of research projects and initiatives to be undertaken around Antarctica, but also to identify the remaining gaps in terms of spatial and temporal coverage, as well as preparing the future proposal and expedition for the future. In terms of milestones, we aim to coordinate multiple oceanographic cruises and sampling efforts, which is quite challenging because usually they are cruises from different countries with different objectives, with different rules and different time. Then we aim to promote as much as we can the exchange of scientists between the different cruises and especially the early career researchers. And then to organize regular workshops during conferences. And then what we aim to do as a SCAR product is first to publish sampling protocols that can be distributed through the SCAR website to the different initiatives that will be led around Antarctica. And if you are not an expert on some sample, on some proxy, for instance, it doesn't prevent you to collect these samples for other groups that will work later on these proxies. Then we aim also to create an online free access data sets in the SCAR database and produce a map including all the measured parameters. So that means we would like to improve also the access of different surface sediment for those who want to work on surface sediment for proxy calibration. And then in the end, we want to have some publication about the inter-calibration of paleoproxies or new paleoclimate records spanning the last 2,000 years, the one that I mentioned a bit earlier in my presentation. So in terms of workshop, we already organized 2 workshops. One was during the Instant Conference in 2023 in Trieste. So we had a half-day workshop there. Then in 2024, last year, August, then we were also organizing a half-day session during the SCAR conference. During the next International Symposium on Antarctic Earth Science, we also aim to organize a workshop. This will be on the Thursday 21st, between 1:30 and 4:30 p.m. And I really encourage you, for those who are attending the symposium, to join. And then we will have also another workshop next year during the SCAR conference in Oslo, Norway, 2026. Now I leave the floor to Dimitri Evangelinos to explain you the different steps of the IcePro project.
Excellent. Thanks, Johan. I'll share my screen as well. Let me see if I can. So, yeah, okay, good. Okay, so do you see my screen now? Do you hear me?
Yes.
Excellent. Yeah. So, hi everyone. I'm Dimitris Evangelinos, and I'm an early career postdoctoral fellow at Imperial College London. I'm a marine geologist and currently co-leader of IcePro together with Joanne Tourneau. So I'm going to give you a brief overview of the IcePro current activities and progress. So currently, we have three main working groups within IcePro. The first one is the diatom working group, which is led by Amy Leventer. We have a DNA working group led by Linda Ambrecht. And we have a third working group on biomarkers led by Bella Duncan. But I also want to highlight that we welcome researchers and especially encourage early career scientists to get in touch with us and get involved and propose a working group on a proxy that they're working and benefit from IcePro coordinating sampling effort and also the data collection we do over the last couple of years. So one of the key and first objectives of each working group was, as John said, to develop a robust sampling protocol. This will ensure to get in an easy way high quality comparable data that we can use it in different expeditions. So this is an example of a protocol produced by the working group on DNA. And as you can see, it's a simple 2 pages protocol that you can easily read and includes all the essential information on sampling DNA, from the chemicals and consumables to the personal protective equipment, such as like glasses and face masks. Also give an idea of the suitable system, coding system or other sampling system to get this kind of data. And of course goes through the main risks of contamination during some sampling and how to avoid them. And also how to store the samples on board in order to have a good preservation and be able, when you go back to the lab, do the analysis. So we have developed similar protocols from all three working groups, and soon will be available in the SCAR website, so then can be easily accessible and people can use them. So in IcePro, we mainly focus our collection on surface seawater, water column water and seawater and surface sediments. And we're interested in several parameters such as phytoplankton species, nutrients, biomarkers, DNA, and other hydrological parameters such as temperature, salinity, fluorescence, etc. And we use mainly the seawater pipeline on research vessels, get the water, surface water, collect it either as it is in plastic vials or filter it. We use the CDD for the water column sampling, phytoplankton nets, and different sediment coring system for getting these undisturbed superficial sediments, including monocorer and multicorer. Here in the left in the picture, you can see, for those that are not familiar with a monocorer, which is this kind of small gravity coring system, like 30-centimeter tube that it's attached with a rope to the frame of the rosette of the CDD and goes together and get the sediments. So this is the sample collection we do. And over the past years, we've been coordinating several sampling efforts on different international Antarctic expeditions. We start this with Spanish teams on board Asperides in 2018, 20, and 24. There were like 3 different expeditions in the Antarctic and the Drake Passage, Antarctic Peninsula, and Bransfield Strait, where we collected surface seawater, water column, and surface sediments. Then in 2019, we collaborated with a South Korean team on board Aron, and then we managed to collect surface seawater samples from a transect starting in Christchurch in New Zealand to the Rossi in the Jambogo research station, and then the way back. And since 2020, we've established new collaboration with private sector and the vessel commandant SARCO. And we had we participated in 3 cruises, and we managed to sample mainly surface seawater, but also some CDDs. And we covered a transect from Christchurch to a little bit of East Antarctica, Rossi, West Antarctica, and then the Drake Passage, as well as a little bit of the Weddell Sea region. So on those 3 different cruises. Now for the upcoming Antarctic expeditions in the next Antarctic season, we've already planned to collect samples on board Hesperides in the Antarctic Peninsula and Bransfield Strait. Seawater samples and sediments will be collected. That will be this Antarctic season, as well as there are 2 more cruises with Commandant Sarko that again we will collect sediments, surface sediments, and seawater samples, and we will cover an area from Ushuaia to Hobart. It's one of the cruise, and the second one, the continuous, was from Hobart to Cape Town. And also we will collect some samples in collaboration with Australian team on board the Investigator. That will be in the Webb's-Head subglacial basin in the Cook and Ninnis continental shelf and slope. And we aim to get surface water samples, water column samples, and sediments. And also in 2027, we'll go in the same region as part of a Polarin project to again collect sediments and seawater samples. Now, as we said before, promoting early career scientist exchange is a key priority in ICEPRO. So over the years, we have supported the participation of 3 master students and 2 PhD students in different international Antarctic expeditions. And we plan to support at least 2 more postdocs in the upcoming cruises. And this is not only an opportunity for the early career scientists to get new skills during the cruises, but also like getting expanding their own professional network and get their own sediments with their own samples and use them for their own research. So this is my last slide, and I want to show you a work in progress. So all this data we're collecting over the years, our goal is to put them in a database, freely accessible. We're working on that to finalize this and upload it in the SCAR website. And it's going to include information about all the different samples we have collected through the years, including the positions, coordinates, the different parameters that have been measured, where are they stored. So then anyone interested can go back, look there, where are the samples, what kind of samples have been collected, and use them for their own research. So yeah, with that, I'd like to thank you for listening, and please feel free to reach out to us, get involved, and we're happy to take questions.
Thank you, Dimitrios. Thank you very much. Are there any questions? You can write it in the chat or...
So can I will ask a query basically. Thank you, John and Dimitris, for giving a very nice idea of what is being done. And the thing in which I was interested was that these are dominantly biological proxies we are talking about, but there are other paleoproxies like minerals and sediment geochemistry, etc., which may also be relevant in terms of correlation, regional correlations. So, do you think there is scope of developing that kind of proxy working group within the framework of IcePro?
Well, actually, like I listed at the beginning, the aim is to develop a proxy calibration for any kind of tools. So that means we are not restricted to biological tracers. It's mostly because it has been initiated by people working on microfossils, on biomarkers, or more recently on DNA. But like Dimitri mentioned a bit earlier, it's really open to anyone who wants to develop his own working group. And I think we try to encourage as much as we can people who are working also on minerals, on grain sizes, on XRF data, on any kind of proxy that can be used also for paleo-records. The thing that for now, no one came to us and said, hey, I want to make this working group. What do you think about this? So that's why every time we organize a workshop or every time we try to promote IcePro, we also try to push a bit the people, especially the early career scientists, who wants to build their own working group under the IcePro umbrella. So we are absolutely open to any kind of proxies. It's just that ourselves, we cannot manage to organize the working group for different proxies, but we would like to invite strongly anyone who wants to be part of it to come and propose his working group on that.
Thank you.
I have a question. Do you, I was looking at the ICEPRO SCAR website. Do you plan or do you already have GIS platform or a layer that people can look at to see where you collected the sample, where you're going to collect it? Or maybe do you plan to use the SCAR facilities for this?
We don't have yet, but we're planning to, next to the database we're building, having like a kind of interactive way that people can see plotted all the positions of the different samples and what kind of samples have been collected. So yeah, we're in discussion with SCAR while we're still working on finalizing the database.
Yeah, because that would be extremely useful, of course, and with the metadata with at least one email address to contact for,
Yes, that's the idea. That's our goal.
I think that once the database will be prepared, even if the database is something alive, because then you always add some new data and so on. The idea would be probably to talk with the Antarctic map viewer people to see if possible, for instance, to add one layer about, where are the samples located, so people can tick the box and see where are the surface sediment, the water samples, the filters, and so on. But we are not at this stage yet. And I think it's mostly because we are preparing a long Antarctic season next year with at least 4 different cruises. So that means I think after next year, we will have a very nice sample collection around Antarctica. And then we will make them available. So for anyone who wants to work on it.
Yeah, thank you. I don't see any question for now from the chat. So you are coming to Punta Arenas?
Yeah.
Yes, we will.
And the site meeting for ICEPRO will be hybrid?
Yes, we always do
So
hybrid meetings. Yeah. So that would be also good way. So either by a simple email you can contact us, or in these kind of workshops in the main conference that that they're always hybrid. You can get in contact and yeah.
Matilde, it will be good to inform the participants about the availability of YouTube of the last talk.
Seminar. Yes, so for all our public, we are recording these seminars, this Geosciences SCAR seminar, and you can find the first one already in the SCAR YouTube channel, and the second one in a few days, and one by one we will add all of them.
Okay, I think if there are no other questions, maybe we can close.
Yes, thank you then. Thanks a lot, Johan Dimitrios, and thanks for listening. And see you in Punta Arenas.
Thank you very
Thank you.
much, and see you next time.
Bye.
Have a good day. Bye-bye.